So we've seen so far that some examples of the simpler version of genetics where single traits are caused by single genes with big effects. And the fact is that that's not really the model that accounts for most genetic inheritance. So obviously genetics is a lot more complex than that. And we'll start getting into some of that here. A nice illustration here of one of the traits that you can look at in humans that's not simply under the control of one or even a few genes. And that would be someone's natural skin tone. So a nice illustration here. This is by an artist who took photographs of people and then matched them to a background that matches their skin color, and just illustrates that we have a really wide variety. And you wouldn't be able to pin this into even 10 different color categories. There's just so many different possible colors for human skin color. And of course, this is the genetic form of human skin color, not affected by tanning, for example. But people are basically genetically programmed to a certain skin color. Um, and we'll see there's some genetic effects that contribute to this. But if you took skin color just as one trait and you wanted to say, this is my phenotype, what is the cause for that phenotype, you'd be looking at quite a few genes that are responsible for the phenotype there. So the topic here relates to what we call non-Mendelian inheritance. So it's useful to refer back to what we would consider as Mendelian inheritance. So Mendelian is an adjective that comes out of the name for Gregor Mendel, this person who basically figured out genetic principles in pea plants. And as we saw in the pea plants, we had a set of traits that were caused by single genes with a dominant allele and a recessive allele. And pretty easy, in retrospect, those are the easier kinds of traits to figure out a genetic cause for. And these bullet points here illustrate the kind of the assumptions or the conditions that would need to be met for genetic inheritance to be Mendelian. So autosomal, we'll get into that. Autosomal refers to chromosomes that are not sex chromosomes, so chromosomes that are equally inherited by males and females. Complete dominance, as I mentioned, so alleles that are dominant over other alleles. Full survival, so no influence of the genetic, the alleles that are being studied on how the offspring survive. One gene with two alleles, genes being independent from each other. We saw cases of looking at two genes at the same time and having those be inherited independently. And then 100% heritability. That's another concept we'll get to later in the course. Heritability is basically how much of the trait is inherited genetically versus, for example, how much is determined by environmental factors. So if we look at fruit flies, you've had some experience with fruit flies up to this point. If you take a cross of a wild type fruit fly by one of these recessive mutant fruit flies, a lot of the fruit fly traits that we look at are Mendelian. And so crossing wild type by a mutant that's even mutated for two genes, an ebony body and sepia eye, having those mutations, those are covered by the dominant wild type alleles, and the offspring of this cross would be 100% wild type offspring. So a few different subcategories of non-Mendelian inheritance, and some of these first ones break the expectation of complete dominance. So this illustration is from your book, this diagram on the right here, complete dominance versus incomplete dominance. So in complete dominance, as we saw, the presence of just one dominant allele will give you the full dominant phenotype. So in this notation, they're using these abbreviations for alleles of A1 and A2 for these different alleles. And that's meant, I think, to kind of reduce some bias of thinking ahead of time that something is dominant. It's not capital or lowercase, it's just 1 and 2. So in full complete dominance, the homozygous individuals, as we've seen, are, let's say that red is the dominant phenotype in this case, then a homozygous individual for the A1 allele is going to be fully red, and then this other allele A2, a homozygous individual for that allele is going to have white petals. And in complete dominance, one of these alleles is fully dominant over the other. So a heterozygous individual looks like one of the homozygous individuals. So maybe easier to think about the red pigmentation being dominant. So having at least one copy of A1, whether you're heterozygous or homozygous, gives you this uh, full red phenotype. And the other one over here shows that it could be possible that this white allele, the A2 allele that causes the white petals, that also could be dominant uh, in a different scenario. And what we call incomplete dominance is where the heterozygous individual falls somewhere in between the phenotype of the homozygous individuals. So homozygous for one allele or homozygous for the other allele. And then the heterozygous individuals are generally, in the examples that I present for this, we have some kind of color difference. So you can kind of think of it as mixing colors. If you mixed red color and white color paint, you would get a pink color of paint. And that's not too far off from what can cause this genetically, where having not as much of that red allele, having half amount of the red allele, actually causes a half amount of the pigment or something less than 100% of the pigment you would find in the homozygous individual. So there are many ways we can look at genetic inheritance. When we look at incomplete dominance, usually I'll do examples that have something to do with color like this. There's some real examples here in snapdragons. And just like in the illustration from the book here, a good way, I think, to treat the alleles for incompletely dominant traits is to kind of avoid that capital letter, lowercase letter bias where one allele is more important than another, more dominant than another. In incomplete dominance, they're sort of equally contributing. Um, so here we're going to use this notation where they have a C for like the color gene. Color gene with the white allele, color gene with the white allele. So homozygous for the white allele produces white petaled flowers. Homozygous for the red allele gives you red petaled flowers. And then a heterozygous individual with a little red contribution and a little white contribution gets these pink flowers. And getting into incomplete dominance and actually everything else we're going to talk about, we've talked about the difference, the relationship between genotype and phenotype. And looking at alleles, if we understand how alleles are present on chromosomes, how they're inherited, and how genotypes contribute to phenotype, all the same rules for following alleles through a genetic cross, those are still present. Those all still apply. And so I'll show you that we can basically map the relationship of genotype to phenotype and otherwise do the Punnett square just like we would normally. So an example here from eggplants, there's known to be some incomplete dominance in eggplant coloration. So on the left here, a case of incomplete dominance where the homozygous for the purple allele is purple, the homozygous for the white allele is white, and the heterozygous is light purple. And you could find a similar pattern, say you did have instead complete dominance, you'd see the same genotypes and just a difference in the phenotype of the heterozygous individual. So looking at this trait, this is still a phenotype caused by one gene. We have only, in this example, two possible alleles, the P purple allele and the W white allele. Every parent gives one of their alleles to the offspring. So we still have gametes, inheritance, Punnett squares, just as before. And the only thing really that's different is how the genotype maps to a phenotype. And so we can draw kind of a table for that. And for every possible genotype, just kind of plan out ahead of time what the phenotype would be. So if you're doing a genetic cross, I think this is the kind of thing that's important to know. Maybe write one of these out for yourself, just so you can refer backwards and forwards between genotypes and phenotypes. So if you know the genotype, then you can solve for phenotype. And if you have a phenotype, you may know or you may not know 100% what the genotype is. So for example, in complete dominance, we saw this before. If you have a purple eggplant and the inheritance is due to complete dominance, you would know that purple is the result of at least one purple allele, but you don't know if it's homozygous or heterozygous. In the incomplete dominance example, this is a little bit of an extra convenience where we have three genotypes, three possible genotypes, and three possible phenotypes. So you can actually go back and forth between genotype and phenotype in this case. And looking at any one phenotype, you would know the genotype and vice versa. So let's do an example here. Again, we're in the incomplete dominance topic, and I've chosen a hypothetical color mixing example. So I chose the organism of sugar gliders. So parents with black fur and white fur produce an intermediate gray fur phenotype. What are the odds that two gray sugar gliders will produce a gray offspring? All right, so for this example, I think it might help to kind of establish the rules. So black by white gives us gray. And I don't remember if I said in the example, but a lot of the examples that I'll give you, of course, would be based on the assumption that we're still looking at a trait caused by one gene. And unless I tell you otherwise, one gene with two alleles. And obviously the phenotypes in question would be due to the alleles present. So first thing I think to do would be maybe to establish the genotypes and the allele indicators that you want to use for these. So we can just do C again for color, and two parents with the black color and the white color respectively would produce an offspring that gets the B black allele from the black parent and the W white allele from the W parent. And that gives us the heterozygous genotype, which gives us the intermediate phenotype. So we've already, I could draw out a table, but it's already on here pretty well. We have three phenotypes and three genotypes. So going back to the example, what are the odds that two gray sugar gliders will produce a gray offspring? So what happens if we do gray by gray? I'm glad you asked. So again, converting our phenotypes straight into genotypes. And we could turn those into a Punnett square. Do the first step here, determining the possible gametes. So everybody's heterozygous. They can produce two kinds of gametes. I like to circle the gametes just to make it a little more clear where the separation is there. So every gamete gets, of course, one allele for the gene. We're only looking at one gene. So in the Punnett square, we can do C to the B or C to the W for each parent. So C to the B, C to the W. And our possible offspring are C, B, C, B, C, B, C, W, C, B, C, W. Keeping my heterozygotes with the just alphabetical order for the allele notation. So those are the same genotype, even though they got there from a different combination. And then here we've got CW, CW. So tracking back to our phenotypes, this would be a black furred individual. This would be a white furred individual. And these would be gray. So the question was, what are the offspring? What are the chances that they would have a gray offspring? So the chance of any one offspring being gray furred is 2 out of 4 equals 1 out of 2 equals 50%. Okay, so you might have probably seen those before in previous examples in other courses. Another complication is that a single gene can have more than 2 alleles. So plenty of cases of this. The spectrum basically ranges from wild type, say fully functional version of the gene, all the way down to one that's completely non-functional. And that's an example here we have in the fruit flies. We have some of our fruit flies that have the white eye mutation. So this is a gene, the white gene. And the W plus allele is the fully functional wild type version. And having at least one of these W plus alleles gives you the wild type phenotype. So that is a dominant allele, gives you the dominant wild type phenotype. But if anybody's missing the W plus, there's a couple of alleles. One of them is kind of a halfway functional allele, and it produces a halfway functional color here. So it's a mutant allele that we call apricot. So white apricot is the W raised to the A allele, and that produces this apricot phenotype. And so anybody who has homozygous for the apricot allele or heterozygous if the other allele is the fully non-functional little W allele, those produce apricot color eyes. And it takes two of these fully non-functional W alleles to produce the white eye color. So here we've got now three alleles for one gene, and three alleles put into diploid combinations gives us this 6 possible genotypes. And so again, with the table being able to track back and forth between genotypes and phenotypes, if you knew your genotype, you could go directly to the phenotype. If you knew your phenotype, you'd be able to rule out, narrow it down to a smaller number of possible genotypes that could cause that. Another example from humans now this time, this is a case called codominance. And codominance is a little different from incomplete dominance. Codominance is a case, really, I don't know, when I was a student, it was like the only time codominance came up was for human blood typing. And I think that's fair. I'm going to keep that tradition alive. So codominance, you can think just of human blood type. And the codominance refers to the fact that in this case, we have three alleles. The alleles here are big I raised to the A power, big I raised to the B, and little I. And the codominance is that A and B can kind of exist together and both contribute to the phenotype. And they're both dominant over the little I allele. So what is the phenotype? Well, the phenotype is blood type. So let me get us a clean slate here. I think this example is really nice for showing the contribution of genes and alleles to phenotype. So if you have a individual that's got these big I alleles, I guess I'll put them both on there, then they produce red blood cells that have antigens on them. So the genetic instructions for the big IA allele are the instructions for how to make this cell identifier, basically. So this is a red blood cell. It's got that kind of donut dimple kind of appearance. Having the big IA gives this A antigen. So antigen is a cell identifier. Having the big IB allele does the same thing, but with a different kind of antigen, the B antigen. So obviously they don't look like this, but you can think of the proteins that they put on them to identify themselves. You can kind of think of those as just advertising the presence of A or B. All right, so of our possible genotypes, let's see, we saw 3 alleles can combine into a few different kinds of genotypes. We got 1, 2, 3, 4, there'll be 6 total. It's possible also to have a combination of big IA and big IB. And here's one of the ways that you can think about them being codominant is that A and B antigens both get made. So the A allele doesn't override the B allele or vice versa. They both get put onto the outside of this cell, and it's basically identifying itself as two things together. So it says, I'm A and B. And then finally, the little I combination produces cells that don't have A or B antigens on them. So the blood type here, I didn't really spell that out before. The blood type for the top one is type A. The type for the second one is type B. The type for the third one is type AB. And the bottom one we call type O, which is just basically none of the above. So for human blood types, there are other ways to have blood type, but this is kind of the most commonly considered blood type: A, B, AB, or O. And what that means for blood typing is very important for if anybody is donating blood, people have to donate their blood to somebody who is a proper recipient for their blood type. So if you are type A and you donate your blood to somebody who's type A, that's just fine, right? Because that person's body has already seen the type A antigen and will accept that, and it doesn't look foreign. But if you were type B and you tried donating to type A, the B antigen is not something that this individual's body has encountered before, and we'll do angry face there. This would actually cause a serious reaction where the blood would attack the foreign blood, and you get blood clotting, and people can die from this. Fortunately, we've known about this for a long time, about blood compatibility, and certainly precautions are taken so that people don't, in a hospital environment, get donated the wrong kind of blood. There are other ways. Try not to mess up our image too much here. Let's say you are donating to somebody who has type AB blood. This individual has experience with the A antigen, has experience with the B antigen, and any of these blood types, A, B, or AB, those are all okay. In fact, O is okay as well. O is what we call the universal donor because it doesn't come up with any of these, we'll call them maybe offensive antigens. One analogy I like is that if you think about sports teams, maybe. So type A has cells that are advertising their love for the A sports team. Type B loves the B sports team. Type AB, they're okay with both sports teams, A or B. And type O doesn't really express a sports team preference. So all of these people would be welcome at the party of somebody who likes team A and team B. So if you like team A, you're welcome. If you like team B, you're welcome. If you like team A and B, you're definitely welcome. And if you're not much into sports, you're welcome as well. But if you have a contrast where somebody likes team B and they encounter someone who likes team A, that's incompatible. And let's say type O over here. So it's type O. Type O doesn't express a love for sports, but really would rather not talk about sports. So I'm getting too many lines in here. So anybody who comes into the type O recipient with an A or B or both antigen is also not welcome. So type O would be an individual who has a hard time receiving blood, would have to be only type O. Somebody who has type AB blood would be able to receive any kind of blood. So they call AB the universal recipient, and they call type O the universal donor, so that people with type O could donate to anybody else with any other blood type. A lot of times I get the question about, what about plus and minus, A positive, B positive, A negative? That's a whole other type of inheritance, and we're not going to cover that here, but we'd be able to look that up if you wanted to. All right, so I basically drew out what is also shown in this image here about compatibility. Shows in this row that Type O can give blood to anybody, and in this column that type AB can receive blood from anybody. And then across the diagonal, every type of blood can give and receive from the same type of blood. All right, so let's see if we can solve a problem here. You can turn these into interesting genetic problems. The children of a man with type AB blood and a woman with type B blood could have which blood types? All right, so if the man is A for the father, and B, so we've got their blood types there, and I've left it pretty open, mostly so we can work through this. We know that type A could be heterozygous or homozygous. So homozygous there, or heterozygous. Type B could be homozygous or heterozygous. And I guess if you want to consider the offspring of this cross, you could consider all possible genotypes for somebody who is type A for one parent and type B for the other parent. So would it be possible to have these two parents produce offspring that have type A? That would be possible if dad donated an big IA, that's quite a bit there, and mom would have to donate one of her little eyes. So she would have to be heterozygous for this to be possible. Same thing in reverse for type B. These two parents could have a type B child if mom gives a big IB and dad gives a little I. And type AB is possible if dad gives an A and mom gives a B. Messing this whole thing up here. There's only one genotype for type AB blood. And type O would be possible if mom and dad are both heterozygous. They would each have a little I allele and they could give that to their offspring. All right, here's a little more complicated example. So let's say that the man with type A and the woman with type B have a child with type O blood. Maybe I can skip this all back. Clean it up. Quick, go back. Awesome. All right, so let's say they have a child who has type O blood. When I write a question like this, I'm basically giving you a new piece of information to narrow down the genotype possibilities. So we know based on that information that dad has to be heterozygous and mom has to be heterozygous. So the only way for them to have type A blood and type B blood respectively, and to have a child who has type O blood, is for them to be heterozygous. Those are their genotypes. That's the rest of the question here. What are the odds their next child will have type A blood? So we turn that into a Punnett square. Dad's got big IA and little I. Mom's got big IB and little I. 1/4 of their offspring possibilities are type AB. 1/4 of the possibilities are type B. 1/4 of the possibilities are type A, and 1/4 of the possibilities are type O. So actually, this set of genotypes could produce all possible human blood types, and the odds of having type A are 1 out of 4. Just one little thing to clarify. The question says that they've already had a child with type O, and you might be inclined to say, well, they've already done that. Maybe that's less chance that their next child will be type O. And that's not true according to probability. So every child is a new roll of the dice, as it were. And so every time it's always the same odds of 1/4 chance of having somebody with type A blood. Just something to keep in mind. Okay, along with other non-Mendelian expectations, so Mendelian traits, as we mentioned, have a feature where everybody survives, but we'll see lots of cases through this semester where individuals can experience negative health effects by inheriting one of the alleles under consideration. So if individuals receive alleles that cause them to die immediately, so let's say as a result of fertilization, a cell is not viable, or an individual who is more likely to die or have less chance of reproducing at some point in its lifetime, all those can have negative effects on the outcome of these genetic crosses. So I guess if we're looking just at the offspring, at a young age of the offspring, we'd probably be interested in uh, alleles that maybe affect survival up until the point of those individuals being counted. So one way these are sometimes referred to as lethal alleles, or we'll see cases of essential genes as well as one of our upcoming topics. So if a gene is really important and necessary, and through inheritance, somebody inherits alleles that don't do that essential function, then that can cause them not to survive. And this kind of happens behind the scenes in genetics, um, and we don't always see the effects of it, except maybe under controlled genetic conditions like this. So here's kind of a convoluted example, but it is a real example from plants. There is a gene here called the PR gene. And the PR gene has a role in putting wax on the epidermis of a plant. And wax is a good way to keep plants from drying out. If they don't have that surface wax, then all their juicy insides are exposed pretty close to where they could evaporate and lose that water. And so there's a form of this over here, this PRPR. Individuals that are homozygous for the recessive allele of the PR gene, they actually don't survive. And they don't survive to the point where you would be able to count them. So it's kind of a quirky question here, but sometimes this comes up as a question. If you were evaluating the offspring of a cross and individuals not surviving was a possibility, you might not get the same ratios as if every offspring individual survives. So let's see here. We got a cross of ratio of mature plants would you count if you bred two bright green plants together? So bright green has this genotype of PR plus PR. So we could do that cross. So it's just a pair of heterozygous individuals. And our Punnett square becomes PR plus or PR for one parent, PR plus or PR for the other parent. And some of those individuals would be homozygous dominant. Some of those individuals would be heterozygous, heterozygous again. And here we've got homozygous recessive. So in this case, we've identified this genotype as one that doesn't survive. So if you were just counting offspring, you might not notice that you were missing these individuals. And so of your mature offspring, you would count 2/3 of them, right? So there's only 3 viable categories, and that was bright green, and what did I call the other one? Glaucus, green gray. Third glaucus. So instead of getting 4 possible outcomes, we have 3 possible viable outcomes, and 2/3 bright green, 1/3 glaucus color. All right, let's see. We had an expectation of genes contributing independently to the phenotype, and that doesn't always hold true. There can be genes that interact in a variety of ways, and one of the ways that they can interact is that one gene, I think maybe a good way to think about this one is a gene being sort of a gatekeeper to a phenotype. So this is an example from Labrador retrievers. There are three main kinds of Labrador retrievers: the black lab, yellow lab, and chocolate lab. And there are two genes in this case that are influencing the coat color. So gene B and gene E, they have dominant and recessive alleles for each. And we can kind of maybe just look at this for a minute and see if you can figure out the rules for how these traits are inherited. So I've got them as genotypes with a blank if it doesn't necessarily matter what's in that spot. So the black lab has at least one capital B and at least one capital E. So at least one dominant allele for each of these genes. The yellow lab doesn't matter what it's got for the B gene. If it has two recessive little E alleles, that's enough to give you a yellow lab in this example. And then the chocolate lab is recessive homozygous for the B gene, but at least one dominant big E allele. So I mentioned gatekeeping. So spell out the causes here. So we have the E gene first. And if the E gene is homozygous recessive, that goes directly to causing the yellow lab coat color. And then the other two coat colors require this first step of having at least one big E allele. So we're sort of branching out into possibilities. If the lab has two little e's, it doesn't matter what the B gene genotype is, it just doesn't get considered here. So we don't have to worry about that one. I was going to say brown, let's do chocolate. So this is the case of epistasis. And epistasis is the literal translation. It's from Greek, and it basically means that one gene is standing on the other one. So the E gene is the gatekeeper and decides or determines whether the B gene will get interpreted. So if there's not a big E allele, then in those individuals, the B gene doesn't get looked at. And like I said, it goes straight to producing a yellow lab. And it takes a combination of two genotypes to give you the black or the chocolate lab coat color. So let's do an example here. We've got a lab in the lab for us to help us with this one. So suppose you cause a black lab and a chocolate lab, and they produce a chocolate lab and a yellow lab. All right. So let's see if we can draw this out for a starter. Black lab by chocolate lab for starter. Boink. All right, so black by chocolate. And let's see, we've determined that each of these need a big E allele because they're not the yellow lab. And then the black lab was at least one big B, and the chocolate lab was homozygous little b. So we know some about their genotypes. We don't know all of it. And their offspring, they have produced some offspring, and they have a chocolate lab and a yellow lab. So I guess I'll split these off into two. Chocolate lab and yellow. So basically both of these are possible from those parents. The yellow lab doesn't, oopsies, in the wrong order. Nope, that was right. Sorry. Yellow lab doesn't matter what it has for the B gene, but it would be homozygous recessive for the E gene. The chocolate lab, as we've seen up top already, it's homozygous recessive for the B gene, and heterozygous or homozygous, but at least one of these big E alleles. So can we use this information to figure out the genotypes of the parents? Think about that for a second. Why, we sure can. So we can look at the B gene and know that we have an offspring that's got little b and little b. So little b from the chocolate lab parent, that's easy enough. But the other parent also has to give us a little b. So that tells us that this individual, the black lab parent, was heterozygous because it had at least one, had to have a little b allele to give. Over here, the fact that these two parents had a yellow lab tells us that they would have to have both a little e allele to be able to produce this homozygous recessive little e yellow lab offspring. Okay, so what are the genotypes of the parents? We done figured them out. So here's another question. What are the odds that the same parents would produce a black lab offspring? Can these parents produce a black lab offspring? Let's see if we can figure out their gametes. Leave some space here. So we could do a gamete square. There's a time saver we can do. There are two little b alleles. This is homozygous for little b. We don't have to do another line for b because they would be the same. Put b's first. So b with little b with big E or little b with little e. The black lab parent has two kinds of b and two kinds of e. So this is our gamete square, big B, big E, big B, little e, little b, big E, little b, little e. All right. Ran out of space before I thought I would. All right, so Punnett square would have our gametes from the chocolate lab parent. I'm bringing these over from there to be across the top. And then I'll bring these in from there to be along the side. That was all four possibilities. So big B with big E, little b with big E, big B with little e, and little b with little e. So keeping our B's together and our E's together. Capital letters before lowercase letters. Okay, what was the chance of producing a black lab offspring? So black lab genotype possibilities are at least one big B and at least one big E. So that fits it, that fits it, that fits it. So this gives us 3 out of 8. Black lab. I guess if you wanted to figure out the rest of this, we've got 2 here with the little e, that's our yellow lab, and then 3 with the at least one big E, wiggly line under those. At least one big E and homozygous recessive little b. That would be 3/8 chocolate lab. So 3/8 black lab, 3/8 chocolate lab, 2/8 yellow lab. Oof. Forgot I had these in here. Why don't I give us a little break here?